Good morning, bon dia, buongiorno, bonjour. Thank you very much for this introduction, Breixo. And it's an amazing opportunity to share this event today with so many colleagues, all of them outstanding experts in their field of research. And it's incredible to be able to learn about so many topics in such a brief period of time. And in this sense, I'm very glad to share with you some of the advances we have made as part of the Atlantic Positive project. And my goal with this talk is to help broaden our vision about this phenomenon of the yellow-legged velutina Asian hornet in our countries by trying to explain some of the impacts that Vespa velutina may have on the prevalence of endo and ectoparasites in apiaries. This result is the collaboration. This study is the result of the collaboration between our group and the group of Santa Rojas and colleagues from the University of Vigo. Both institutions are located in Galicia, the northwestern corner of the Iberian Peninsula. More specifically, our group is part of the Universidad de Santiago de Compostela, where our group has been studying parasite genetics for many years and is specialized in parasites from honeybees, wasps, and other Hymenoptera species. Many studies have indicate that Vespa velutina's predation pressure consequences are directly or indirectly some of the factors associated with high interannual death of domestic honeybee colonies. And the fourth, this isn't the, wait a second, there's a problem with the slides. Sorry for this technical problem. I'm back in the game. As I was saying, both institutions, both our group and the group of Santa Rojas, are located in Galicia, in the northwestern corner of the Peninsula Ibérica. And more specifically, our group is part of the Universidad de Santiago de Compostela, where our group has been studying parasite genetics for many years and specialized in parasites of, as I said, honeybees, wasps, and other Hymenoptera species. In many studies, there's this indication that Vespa velutina's predation pressures consequences are directly or indirectly some of the factors associated with a higher internal death of domestic honeybee colonies. First, these hornets hunt and feed on bees, which directly produces a reduction in the effect of a reduction in the worker power with the consequences this has. Secondly, it has been reported how the attacks of Vespa velutina near hive entrances also reduce foraging paralysis. And kind of as a novelty, we propose that Vespa velutina also induces shifts in colony dynamics, like individual and social hygiene habits. By this, I mean all these changes may affect the group immunity, that is a type of immunity that relies on social immune processes developed by eusocial insects that include the removal of infected nest mates, self-removal when infected, depositing corpses outside the foraging ranges, collecting antiseptic saps, and behavioral fever, that is when where adults sufficiently raise the temperature of the colony to combat brood infections, and antiseptic enzymatic secretions, among others. And group living is favorable to pathogen spread due to the increased risk of transmission among individuals. So our hypothesis is that the presence of Vespa velutina in front of the hive triggers changes in honeybee colonies ethology, which has negative repercussions on the social group immunity and therefore the health of the entire colony. The foraging paralysis that Vespa velutina induces brings on density-dependent alterations in the prevalence of parasites and the lack of hygienic habits that weakens the colony and predisposes it to failure along the winter. To test this hypothesis, we compared the prevalence of ecto and endoparasites, more specifically Varroa destructor, Nosema ceranae, and Nosema apis, in two types of honeybee colonies. Some of them were protected with electric halves designed to reduce the predation of Vespa velutina, and some were not. So what we did was to measure not only the parasitism, but among others, we also registered the predation pressure of Vespa velutina for each hive, the foraging activity of the colonies, and the rate of overwintering survival. To collect these data, we have been collaborating with Sandra Rojas and her team from the Vigo by sharing experimental apiaries. And for this purpose, three apiaries were designed. They are located in the south of Galicia, next to the border with Portugal, where Vespa velutina is pretty much present everywhere and quite abundantly, I would say. Each apiary is a biological replica, as the colonies come from the same breeder, are from the same age, etc., which consists in each apiary consists in a group of 6 hives that are protected by an electric harp, and another group of 6 that remained unprotected, as shows the scheme shown on the slide on the left. The harps are specially designed to selectively capture hornets that roam around the hive while looking for potential prey. Due to the wingspan of the velutina, they touch 2 electrified cables at the same time and receive an electric shock that stuns them for a moment and make them fall into a trap underneath. This method is harmless for smaller insects like bees, and because we wanted to test if these protective tools can have a positive effect on the sanitary state, I'm pretty sure that Sandra will be able to explain better the design of the harps and the apiaries in the following speech she's given right after me, in the case there's any doubt. To quantify the presence of hunting Vespa velutina in front of hives' entrances, as well as the foraging activity, we used video recordings. A camera was placed half a meter in front of the hive for lapses of 10 minutes, and these recordings were performed during the midday. This was done monthly from July to October, and that is previous to the intense attack of Vespa velutina and after. The resulting videos were visually analyzed later in the laboratory, both manually and by using a software developed by Friat and Gamba called Boris, which is a behavioral observational research interactive software. The selected image for this slide is a screenshot of one of those video recordings, and there should be plenty of bees working. Quite surprisingly, as you see in the picture, there are only 2 bees present. And on the right, you can see the cause of why this is happening, which is a hunting velutina. Regarding the presence of parasite species, in the case of Varroa, it was done following a standard method for Varroa quantification, and in the case of Nosema, it was done via molecular methods of PCR detection. The bee sampling was done both at the beginning of the experiment in July and at the end in October of the years 2020 and 2021. Regarding the predation pressure of Vespa velutina, we obtained the following results. Here in the graphic, we have a panel that is divided in 3 parts, one for each apiary. Each sub-panel displays the evolution of the hunting pressure showed in the y-axis along the month that lasted the experiment: July, October, August, September, and October. The red lines represent the unprotected hives, while the blue ones represent the protected ones. As we can see, generally the blue ones, the protected hives, are less attacked than the ones than the hives that remained unprotected. In the first case, in this case, in the first apiary in green, we can see how a high predation situation scaled from July to August, that dropped in September, but increased again in October. This same pattern was observed in the second apiary that had an intermediate hunting pressure, and this drop in September can be explained to harsh climatic conditions. In the case of the third apiary, the tendency was always increasing, although it was classified as a low-intensity apiary regarding velutina attack. As a result, a strong variation in colony dynamics depending on the treatment and the site was found. As we can see in this sense, here we can see the evolution of the foraging activity of each set of colonies, expressed as the number of bees counted in the hive entrances in a period of 5 minutes. in the site with the highest hunting pressure, which is the first one in green, we observed a similar response in both type of hives. Again, the red ones are the ones that were unprotected, and the blue ones the ones that were protected. And they both gradually paralyzed their foraging activity as the hornet season went on, and although they kept some residual activity. Meanwhile, in the case of the intermediate velutina pressure apiary, the levels of foraging activity also kind of paralyzed when the velutina was at their highest. Nevertheless, in this case, in October, the group of hives that were protected recovered their foraging activity at the end of velutina season in October. And in the case of the apiary with the lowest hunting pressure, the one surrounded in blue, both treatments were kind of similar. The foraging activity was at their lowest when velutina abundance was at their highest. But slowly they recovered after the predation pressure was started decreasing. In the end, we combined all these variables, both the predation level of Vespa velutina per hive, the foraging activity, winter survival, and varroa and endoparasites, to get an actual image of this trophic interaction and the protection that electric harps can offer offered in our experimental apiaries. I will now present the results that we got by now of the prevalence of the ectoparasite Varroa destructor, and by using a standard method of estimation of Varroa, infestation was measured previous to the attack of velutina, and where we found no statistical differences between neither the sites or treatments as expected, because all of the hives came from the same breeder, had the same origin and age, etc. Here in this slide we have the results of October 2020. In the y-axis we can see the number of Varroa per each 100 bees, and in the bottom axis we can distinguish each 3 sites ordered from higher to intermediate and lower hunting pressure. In the case of the scenario with the highest attack, Varroa infestation was 13% higher in the protected hives. This is similar to the low pressure hives, where the increase was around 11%. Nevertheless, the hives with intermediate attack, this pattern does not occur. Also, for each individual hive, there is no association between the data before and after the attack, meaning that the most infested hives after the attack before the attack are necessarily the ones that have the most Varroa after the attack. So, at least with the current data, we cannot stand that electric harps are enough to prevent Vespa velutina from directly affecting Varroa, but nevertheless, this can have many reasons, as the fact of the hive being quite young. This was their first year, so there was not too much overcrowding once they were paralyzed, or the fact that Vespa velutina pressure wasn't high enough in 2 of the apiaries that year, among others that I can comment later. We have found, notwithstanding that when data were analyzed individually for each colony, that there is a slight positive correlation between the number of Varroa and foraging paralysis. Regarding the endoparasite, the presence of endoparasite in the digestive systems of these Apis mellifera, we have analyzed the 2 most relevant Nosema species, which are Nosema cerane and Nosema apis, for samples. And here we can see the results, the preliminary results for the sampling of autumn 2020. In this graph represents the percentage of individual honeybees that contained detectable Nosema DNA in their digestive systems. We have the colored bars, which represent Nosema cerane, and on the top we have the percentage of samples positive for Nosema apis. As in the previous figure, we have the highest level in green on the left, and in this case, the light bars indicate the results of the hive that were protected, and the darker ones the results of the hives that were unprotected. The light green bar indicates 40% of positivity, taking account both species of Nosema in protected hives, and the dark green one shows 32% of positivity. In the case of in the apiary with intermediate hunting pressure, we saw an almost 5% increase of Nosema in the unprotected hives, while in the lower hunting pressure apiary, there was a 25% increase of Nosema in the hives, comparing the hives and in the treatment with the unprotected hives. Although no statistical difference was observed, there is a tendency of these results which is consistent with the expected results, considering their colony dynamics. Because in both cases, with the highest and intermediate Vespa velutina abundance, these first two, almost all the colony paralyzed their foraging activity during the summer in both treatments. But in this case, where the velutina abundance was the highest, neither of both treatments recovered their foraging activity. So we expect very similar results. While in this apiary, which had an intermediate hunting pressure, the protected hives recovered their foraging activity, so there should be, we expected, less quantity of Nosema. Although with this current data, we cannot say that we can say that there is a slight effectiveness of the hives in scenarios with a low and intermediate predation pressure. And the more frequent the attacks of Vespa velutina, the more diluted this protection of the hives gets due to the similar pattern of foraging activity between both treatments. Posterior analysis of individual hives showed a positive correlation between Nosema and nosemosis and the colony dynamics. And we have seen that at a high predation pressure, the more foraging paralysis there is, and a higher prevalence of Nosema species. Just to sum up, we have observed profound effects of early foraging paralysis in our experimental apiaries, but how did Vespa velutina affect this group immunity and the level of parasitism in each colony? It's important to emphasize that these results are preliminary, as the 2021 samples have been recently collected and are just in the moment being processed. So, but nevertheless, the global data from 2020 shows that there may be in fact a detrimental effect of Vespa velutina attacks on honeybee colonies' health by interfering their social group immunity. And when taking the data globally, there is more attack in the more attacked a colony is, the higher prevalence of Varroa and Nosema it shows. Also, individual hive data analysis shows that there is a strong correlation of nosemosis and Varroa presence in highly attacked, unprotected hives. This points out that Vespa velutina may be indirectly causing both types of an increase in both types of parasites, although we should maybe not expect the same patterns in both types of parasites, as their method of infestation and expansion varies. Varroa is quite endogamic and depends on brooding quality and quantity, while Nosema may be more related to more dependent on hygienic habits and high density situations. We expect to have more consistent tendencies once we got all our data together from both years, especially because this effect is expected to be more noticeable in the hives that survived this year, as they are older and more massified. And yeah, so we can say that Vespa velutina might, via group immunity disturbance, affect honeybees' health by triggering a high prevalence of ecto and endoparasites, a higher prevalence. And we have seen higher varroa and nosema in highly attacked hives, and also a correlation of Vespa velutina predation pressure, foraging paralysis, and varroa in the case of highly attacked hives. We have seen that adapted beekeeping management techniques may reduce Vespa velutina impact, as electric harps do have a positive effect on honeybee general health regarding parasitism, and by avoiding colony collapse meaning the winter colony survival, that we found that winter colony survival correlates with harp protection, and found that harps can increase up to 40% this overwintering. And although there is a lot of research to be assessed. And thank you very much. I hope I explained myself the best, and sorry for the technical problem. And if I would be more than happy to get more into detail, and if there's any question. Thank you.